Good afternoon. My name is Michael Maguire. I'm CEO of Avectas. I'm delighted to be speaking at the Cell meeting on the Mesa and looking forward to introducing you to our cell engineering platform, SOLUPORE. Avectas is an Irish company. We're approximately 10 years old. We've 35 staff located outside of Dublin. They're in process development, engineering, and some administrative roles. We've a further 3 staff based in Cambridge, Mass. SOLUPORE is a non-viral, non-electroporation cell engineering technology. We've raised $45 million in capital to date. We've an experienced leadership team, board of directors, and a world-class scientific advisory board. Our collective mission is to commercialize the SOLUPORE platform to enable next-generation cell therapies. SOLUPORE is a physicochemical transfection process, and it enables complex multi-step editing typical of next-generation cell therapy products. On the left-hand side, we have a single-use transfection chamber. Cells are introduced into this chamber in media at normal physiological concentrations. The media is removed through a highly engineered filter, and the cells are then precision sprayed with a solution which contains both the cargoes and a permeabilization chemistry. The membrane is transiently permeabilized, enabling the cargo to pass through into the cell. And when this step is complete, the cells are immediately resuspended and can go on to the next part of the manufacturing process, be that proliferation or be it differentiation. We've published the underlying mechanism and in +1 in the past, and more recently, we've published applications of the technology in cytotherapy in 2021. So in this presentation, I want to speak about the benefits to partners of using SOLUPORE technology. I want to speak to unprecedented flexibility in complex editing. I want to speak to the health and phenotype of the cells which come through the SOLUPORE process. Critically, I want to speak about the functionality of cells, the cells which will go back into the patient as a medicine have to be highly functional to be effective. I'll speak about our regulatory strategy and pathway to GMP, and lastly about ease of adoption. How do partners get their hands on SOLUPORE initially to start incorporating SOLUPORE technology into their product development and manufacturing processes? So taking that last point first, ease of use and ease of adoption. So we've developed the research tool. The research tool is a compact version of the SOLUPORE platform. It's open, it's non-GMP, it operates inside of a biosafety cabinet, but it doesn't take up much room. It can be taken in and out for a particular experimental session. The device has onboard guided workflows which help users get going very easily, very simply with the process. And there is ample SOPs and other support documentation. There's also our team who have developed the product, and they're passionate about supporting partners in their first use of SOLUPORE technology. And they're really good at it as well. We have a number of these devices with partners globally, and the research tool is the stepping stone to the clinical system. This is the SOLUPORE single-use system, or the clinically aligned system. It's a closed, GMP-aligned, automated, software-controlled system. It's designed specifically for complex gene editing and complex cell modification processes. It operates with a wide variety of cargoes to be delivered to cells, messenger RNA, RNPs, CRISPR-Cas9 plasmids and so forth. And we've done the bulk of our developmental work using human primary T cells and T cell subpopulations, but we're expanding that out to other cell types and look forward to updating the market in due course on that. In terms of commercialization, access to this technology will be under development and licensing agreements. In terms of regulatory pathway, there are a number of aspects to the system, to the SOLUPORE system. There's a single-use consumable, there is the controller, and there is the device, the reagent. We met with the FDA in March 2021 and laid out for our device classification meeting. And in the period which since then we have developed and continue to develop a suite of documentation to support partners with their IND filings, either through a DMF or to make the documentation available for direct inclusion in the partner's IND. That suite of documentation will be ready during the course or towards the back end of 2023. So I'm going to show a number of studies here highlighting the capability of the SOLUPORE system and the flexibility in complex editing. In this first study, we're showing triple edited T cells in comparison with nucleofection, electroporation, in this case, nucleofection. So in the left-hand figure, we're looking at yield of triple edited cells. The edits are common T cell edits that you would associate, they're knockouts that you would associate with immuno-oncology. And what we can see is equivalent yield of triple edited cells. On the right-hand side, however, the SOLUPORE cells have greater functionality. In this case, we're measuring functionality using interferon gamma as a marker, and the response of the edited cells post-stimulation is far greater. The interferon gamma is far greater in the SOLUPORE cells, indicating that the cells are much more highly functional post-stimulation. So in triple edited cells, SOLUPORE cells demonstrate similar cell yield of edited cells, but with superior cell function. So in this next study, once again, addressing sequential editing, on the left-hand figure, we're seeing an edit. In this case, it's a single transfection of a multiplex triple edit, 3 targets, TRAC, CD7, and B2M. And we see initially in the first transfection, there is just north of 40%. 24 hours later, we transfect those cells again, and what we see is a near doubling of the transfection efficiency by going in again 24 hours later. However, on the right-hand side, we can see that the viability of the cells is not impacted at all with that second transfection. So it highlights that sequential editing is possible without an impact on the viability of the cells which have been transfected again 24 hours later. This will provide key unprecedented flexibility, this ability to sequentially edit, and it's a data set that we will continue to build out and be interested in speaking with partners about. Turning to cell health, on the left-hand side of this figure, we can see that the SOLUPORE cells have less early and late apoptotic cells than the electroporated cells. These cells are triple edited cells, and the cell health is better. The SOLUPORE is less toxic to the cells. On the right-hand side of the figure, we can see that the proliferation of those cells is also better than the proliferation of the electroporated cells. And you get more divisions in 24 hours with the SOLUPORE cells than you do with the nucleofection or electroporation cells. So SOLUPORE is less toxic and results in greater proliferation of triple transfected cells, triple edited cells. This next study, we're speaking to cell phenotype. So there's a key interest in the T stem central memory effector, the stem central memory cytotoxic effector cell population. And in this study, we're looking at day 1 and day 4 and comparing electroporation and soluporation. And what we can see is that very important stem central memory subpopulation of the cells is maintained at day 1 and at day 4, whereas the electroporated cells, that population is not maintained. And then showing functionality of cells in this in vitro study. So in this study across a range of cytokines, interferon gamma, granzyme A, granzyme B, and so forth, we show that the SOLUPORE modified cells are more highly functional. They exhibit higher levels of these cytokines, which are critical to their application. So the cells are more functional as measured in this in vitro study. So this next study is a study that we published in Cytotherapy, and it's not a gene editing study. It's a transient mRNA coding for a CAR. But the reason that I've looped back and returned to this study is we saw at the time this study was done, we saw at the highest level of cells, of CAR T cells introduced into mice with SOLUPORE, compared with the equivalent electroporated cells, we saw better functionality of those cells through near complete tumor clearance in several of the mice in this study. And when we think about the preceding studies and the better cell health, better phenotype, and better functionality, this makes even more sense in the light of that more recent data. And then on the right-hand side of this panel, we can see again the higher engraftment of SOLUPORE cells relative to electroporated cells. So in summary, there's a unique opportunity to partner with Avectas now. Both our device formats, the research tool and the SUS, are tech transfer ready. We have a defined regulatory pathway, and we have a highly differentiated technology which will enable complex editing of cells, where the cells that are edited are highly functional and have superior cell health. My colleagues and I are at the meeting all this week. We look forward to meeting with you and discussing how SOLUPORE can be used in the context of your developmental products. Thank you very much indeed.